Hello, I am Anamika Patel, a third year BTech student in Engineering Physics at IIT Roorkee. I will be presenting my graded project titled Brain Age Prediction using MRI data. This project focuses on using machine learning models to estimate a person's brain age from MRI derived structural features. The idea is to use neuroimaging data and computational models to detect whether the brain is aging faster or slower than the person's chronological age, which can indicate early neurological decline. Now, I will discuss the motivation and objective behind this project. Uh, as you can see, on this slide, the human brain undergoes measurable structural changes as we age, such as shrinkage of gray matter and increase in cerebrospinal fluid volume. These subtle changes can be captured through MRI scans, and studying them help us understand normal and abnormal brain aging. Now, predicting brain age is important if because if a model estimates the brain's age to be much older than the person's actual age, it can signal potential risk for neurodegenerative diseases like Alzheimer's or Parkinson's. On the other hand, if the predicted brain age is younger than the actual age, it can indicate good brain health or cognitive resilience. This difference between predicted age minus chronological age is called the brain age gap. If this gap is close to zero, the brain is aging normally. If the gap is positive and large, it means accelerated aging. If negative, it suggests slower or healthier aging. So, the goal of my project is to predict chronological age using MRI derived structural features and to evaluate how well the model generalizes to unseen test data. The dataset used for this project comes from an OpenVHV site. OpenVHV stands for Open Big Healthy Brains. Uh, it contains multiple split train internal test or external test datasets. There were 3,227 samples in the training split, 362 samples in the internal test, and 395 samples in the external test split. Each record includes MRI-derived measurements such as TIV, total intracranial volume, intracranial volume, CSFV, cerebrospinal fluid volume, GMV, gray matter volume, WMV, white matter volume, along with the acquisition setting, magnetic field strength, and the site information. These features represents the structural composition of the brain, which tightly correlates with biological aging. Like this is the snapshot of the data set. The columns includes a participant ID, split, sex, age, diagnosis, TIV, CSFV, GMV, WMV, magnetic field strength, acquisition setting, and site. Here two columns, the split and the sex columns are for categorical, and the participant ID is of I guess no use, and other columns are the numerical ones. The next step is of data pre-processing. In this step, I focus on preparing the raw data for model training. First, I drop the irrelevant columns like participant ID that has no use in the prediction of the brain age, and the split column because like We don't have nothing to do with the split column. Since they both column don't contribute to prediction, I dropped these columns. Then I identified categorical features such as sex, diagnosis, accusation setting, and site. These were encoded using label encoder, which converts text labels into numerical values. Uh, uh, for numerical features like TIV, GMV, and WMV, I used a standard scalar to normalize their range. This ensures all features have zero mean and unit variance, helping models like ridge regression converge efficiently. As you can see, pre-processing ensures that all features are in comparable form before entering the machine learning pipeline. The two columns of participant ID and splits are dropped, and the sex column and the diagnosis columns are converted into 0 and 1. The data was split into three parts: the train, the internal test, and the external test. That next part is principal component analysis. To handle possible feature redundancy, I applied principal component analysis for dimensionality reduction. This step helps capture the majority of the variance in fewer dimensions, making the model faster and less prone to overfitting. As you can see the graph on the slide, it plots the cumulative explained variance versus the number of components. The curve shows that with about 5 components, we can retain almost all the meaningful variance in the dataset. Hence, I chose the top 5 principal components for further training. Ridge regression model, the RCB. This was my first predictive model, ridge regression with cross validation or ridge CV. Ridge regression is a linear model that adds an L2 regularization penalty to prevent overfitting, which is especially useful when features are correlated. Uh, it minimizes the following cost: the minimum of mod of pi minus x beta square plus alpha beta square. The penalty term alpha controls model complexity and prevents overfitting. I tune the hyperparameter alpha automatically using cross validation over a log scaled range from 10 to the power minus 3 to 10 to the power 4. From 10 to the power minus 3 to 10 to the power 4. After training, I evaluated the model on both internal and external test splits. The matrices used were MAE, mean absolute error, measures average absolute difference between predicted and actual ages. R square score measures how much of the variance in true age is explained by the model. These matrices provide a balanced view of both accuracy and generalization. Here is the result of the ridge regression model. The MAE obtained on the trained data was 5.74, on internal test was 5.889, on external test was 5.123. The best alpha obtained was 13.895, the validation MAE 5.691, and validation R square score 0.697. Next, I used an SG boost regressor, which is a gradient boosting framework widely known for its strong performance on tabular data. It builds an ensemble of decision trees iteratively, where each new tree tries to correct the errors of previous ones. This helps capture non-linear patterns that a simple linear model might miss. I trained it with 500 estimators, a landing rate of 0.05, and a maximum depth of 5. Training was done on the same features to allow fair comparison with RISC-CV. As I will show later, the RISC-CV performed better, surprisingly well given its simplicity, while SG would slightly improve results on the internal split. The SG results are the MAE of 2.05 on trained data, R square of 0.968. On internal test, MAE was 4.039 and R square was 0.748. On external test, MAE was 4.843 and R square score was 0.619. To further improve prediction robustness, I also built a stacked ensemble model combining ridge regression, random forest, and XGBoost as base learners. Their predictions were fed into a linear regression meta model, which learned how to best combine the strengths of all three models. As seen in the scatter plot here, each point represents a subject. The x-axis shows their actual brain age. The y-axis shows the predicted brain age by the ensemble. The closer the points are to the right dashed line, the line of y is equal to x, the more accurate the predictions. The ensemble achieved a mean absolute error of 4.7 years and a R square of 0.7, indicating a strong correlation between predicted and actual ages. This demonstrates how ensembling can reduce bias and variance simultaneously. Model comparisons and test predictions. Combined evaluation matrices from receiver and attribute models. Matrices compared across trained internal test and external test splits. Generated sample prediction for the test split included actual age, predicted age, and absolute error for both models. Here is the attached snapshot of the actual age, predicted age by the ridge model, predicted age by the SG boost model, and the absolute error given by both the models. And the second snapshot was of the MAE and the R square for both the models. The SG boost model has relatively lower MAE, but a relatively higher R square. Model performance visualization. Model predictions were compared with actual brain ages across the training, internal test, and external test splits. Scatter plots were used to visualize the relationship between actual and predicted brain ages. A close alignment of points along the right diagonal line indicates strong predictive accuracy. Array distributions were analyzed using histograms uh, to observe the spread of absolute prediction errors, and most errors were concentrated around zero, demonstrating consistent model performance with limited large deviations. This slide shows two types of plots, uh, scatter plot and the actual versus scatter plot of actual versus predicted age. Each point represents one sample. Points near the rightest diagonal lines are well predicted, where points near the rightest diagonal line are well predicted, whereas those for far away represent higher errors. The second one is histogram for the error distribution. This shows how prediction errors are spread across samples. As you can see, most errors cluster close to zero, forming a narrow well-shaped curve. Indicates that the model predictions are unbiased on average. These two plots that in the slides are for the train split. These two plots are for the internal split. As we can see, the in the internal test, the points are more far away than from the right test line than the train data, and this is for the external test. In this line, we can see a relatively closer points to the right test line. Next part is model performance visualization. Performance matrices were computed for all data splits, the train, internal test, and external test. Matrices used for evaluation were mean absolute error, which measures the average deviation between predicted and actual brain age, R score, which indicates how well the model explains the variance in brain age. Mean error shows the average bias. Positive means overestimation, negative means underestimation. The model achieved low MAE and high R square on all splits, confirming a strong generalization capability. Similar performance across internal and external tests demonstrates model robustness on unseen data. As we can see, for the train, MAE was 5.714, for internal test, MAE was 5.889, and external test, uh, 5.123. And the R square for train was 0.679, for internal test 0.586, for external test 0.654. The external test has the lowest MAE and the lowest no, the internal test has lowest R square, and the external test has a negative mean error, which indicates the underestimation. Actual versus predicted age and error analysis. A function was implemented to generate detailed tables for each data split, train, validation, and test. Each table includes actual age, the ground truth values from the data set, predicted age, models estimated values after PCA transformation, error, the predicted minus actual age, deviation of the model's prediction from the actual age. The process involves applying the trained scalar and PCA on input features, using the trained regression model to predict brain age, completing the prediction error for performance interpretation. This step enables identification of over or underestimation patterns across samples and help assess model generalization equality. This is the snapshot of the output. There are three columns: the actual age, predicted age, and the error. The difference between the predicted and actual age. This is a prediction error distribution curve for the train split. The y-axis is of counts and the x-axis is of prediction error, the predicted minus actual age. This is the curve of the error versus actual age for the train split. The x-axis is actual age and the y-axis is prediction error. This is the scatter plot of actual versus predicted age on train split. Each point represents one sample. Points near the right test line are well predicted, whereas those far away represents higher errors. Model evaluation on external test data. External test data used to assess model generalization on unseen subjects, generated and analyzed predicted age, actual age, and error values, visualized results through error distribution plots to detect bias or skew, actual versus predicted age plots to assess prediction alignment, quantitative matrices computed for mean absolute error, which measures average, as I already told you that MAE measures average prediction deviation, R square score indicates goodness of fit, and the mean error reveals systematic over or underestimation of the model. This graph is shown of the the histogram is shown of the external test. The x-axis is of the prediction error and the y-axis is of the count. Here are the scatter plot. The On the top it is of the actual age versus error. On the y-axis there is error and on the x-axis there is actual age. On the right, scatter plot is of actual versus predicted age on the external data set. The most important evaluation in this project are MAE of 5.123 years, R square score of 0.654, and mean error of minus 2.245. The slight negative mean error suggests that the model tends to slightly uh, the slight negative the negative mean error suggests that the model tends to slightly underestimate age for some individuals. However, an R square score of 0.65 shows that more than 65% of the variance in brain age is captured by the model, which is quite good for biomedical data. As you can see from the scatter and histogram plots, from this scatter plot, most points align close to the ideal line, and the error distribution remains and in the histogram, the error distribution remains centered near zero, confirming stable predictions. In conclusion, to conclude, this project successfully built a machine learning pipeline capable of predicting brain age using MRI derived structural features. Key achievements was I built and compared multiple models, which CV, XGBoost, and an ensemble model. Achieved a final MAE of 5.12 years and R square score of 0.65 on unseen test data. Demonstrated consistent performance across all splits. This indicates that the model generalizes well and captures important biological aging patterns. In the future, integrating more advanced models like convolutional neural networks directly on MRI scans could improve accuracy further. So, thank you.